Thanks for joining us for label-free protein assays for process development, kinetic characterization, protein folding, and crystallization screens. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. Label-free techniques for detecting and quantitating biomolecular interactions greatly facilitate such analyses by eliminating the need for labeling reagents and providing significant time savings and flexibility. Layer Interferometry extends the capabilities of label-free analysis further, allowing crude samples, sample volumes as low as 4 microliters, and high-throughput analysis in applications including protein-protein binding, quantitation, affinity, and kinetics. The three speakers today, Marin van der Noot-Kolschotten, Mark Fisher, and Jimin Yu, will share their experiences and insights in just a minute. Before we get started, though, I want to encourage you to submit questions for our panelists. We'll answer as many as we can after the final presentation. Simply type your question into the Ask a Question box and hit Submit. Marin is a senior scientist at Crucell. He uses BLI technology to improve the selection of therapeutic mAbs against viral targets. He uses the Octet system for both quantitative and qualitative applications, including protein A quantitation, binding kinetics, competition experiments, and sometimes even mechanism of action experiments. Marin, we're ready when you are.
Thank you, Tamlin. Of course, I would also like to thank ForteBio for sponsoring this webinar. In this webinar, I would like to present some examples on how the Octet supports the research activities within the Antibody Discovery Department of Crucell. Next slide. Let's start with a brief introduction to the Layer Interferometry technology, BLI in short, which is used in the Octet instruments. BLI is a relatively new technology that can be used to measure label-free molecular interactions. This technology uses the interference pattern of reflected light to detect the binding of molecules to a sensor, which consists of a biocompatible surface attached to an optical layer. This sensor is attached to the tip of a disposable optic fiber to which a light beam is sent. The shift in wavelength explains why the final readout for binding is expressed in nanometers. Next slide. For a better understanding of the Octet working principle, the inside of an Octet red system is depicted. Here you can see the optics box with 8 channels, which allows measurement of 8 parallel sensors. The optics box is movable and can move between the sensor tray and the sample plate, which is positioned on a temperature-controlled orbital shaker. Importantly, no fluidic flows are used in the Octet. These features make the Octet very suitable for both quantitative and kinetic applications. Next slide. Currently, ForteBio has 2 series of Octet instruments on the market, the RED and the QAE series, which differ mainly in sensitivity. In each series, a 384 model is available, containing an optics box with 16 channels for high-throughput applications. Still growing list of disposable sensor types are available by ForteBio as depicted below. The majority of ready-to-use sensors are pre-coded with molecules specific for antibodies, such as anti-human or murine Fc IgG or protein A or G. Also, sensors specific for His or GST tags are available. More general applications, stratified mean reactive or hydrophobic APS sensors can be used. Next slide. At different stages along the road from discovery to product, the Octet may be useful. In our department, the Octet applications for certain technologies, such as phase display or pharmacodynamics, are under evaluation. Especially for the early characterization of antibody characterization, several binding assays are successfully being used, and some of these will be presented in this webinar. Next slide. First, I would like to start with an example of the application of the IgG concentration assay using protein A sensors, which is a fast and easy alternative for ELISA or HPLC-based methods. This assay is based on the binding rates at which IgG binds to protein A. Measured binding rates of unknown IgG samples are in the end compared with rates obtained from IgG standard dilutions. Next slide. In this slide, binding curves are depicted, obtained from dilutions of standard IgG samples. The Octet protein A assay makes use of the fact that binding rates are proportional to the concentration of IgG. From the measured binding curves, the quantitation software of the Octet calculates the binding rates for each IgG concentration, which are subsequently plotted against the IgG concentration to obtain a calibration curve. Next slide. Typical standard curve is shown in this slide, which is used to measure accurately IgG concentrations in unknown samples. Interestingly, the range of such a calibration curve is rather broad, and moreover, the same curve can be used again in the next assay, provided that used dilution buffers are same. Next slide. The Octet-based IgG quantitation assay has been evaluated in our cell line development department. The main objective of our cell line development department is to generate high-performing IgG-producing cell lines suitable for process development. Next slide. In this slide, an overview is shown of the general outline of the generation of a stable cell line, starting with the transfection of Percy6 cells under serum-free conditions. Crucial for this process is a limiting dilution step in which more than 600 wells are seeded, 96 wells, after which 600 wells will, with cell growth, need to be expanded to a larger T25 format to measure IgG production. Unfortunately, volume and concentration in the 96 well format are too low to be detected in the protein A HPLC method that was routinely used at that time. Therefore, it was decided to evaluate whether the Octet protein A assay could already be used in the 96 well stage. But before this evaluation was started, it was first tested whether the HPLC and Octet-based IgG assays would give the same results. Next slide. For this reason, all kinds of IgG-containing samples, low and high concentrations, were tested, both assays, and the results were plotted. As you can see, the IgG quantitation correlated very well. Next slide. Next, the IgG expression of all clones was tested both in the 96 well format using the Octet, as well as in the T25 format using the HPLC method. Based on the data, the deselection criterion for the Octet was set at 22.5 milligram per liter. Using this cutoff value, only a few clones would have been missed that would have met the selection criterion set for the T25 culture format, and therefore it was decided to implement the Octet protein A assay in the 96 well format. Next slide. In conclusion, we found a correlation between the volumetric Octet assay and production per cell in the HPLC assay. Only 3% of clones that would have been selected in the T25 format were missed by using early Octet screening with high deselection criteria, 22.5 milligram per liter. About 400 clones of the total of 600 were deselected and did not need to be subcultured or expanded larger volume, saving at least 60% of resources in this step. So the deselection of clones in the 96 well format using the Octet quantitation assay has now been implemented. Next slide. Short summary on the IgG quantitation assay. IgG quantitation on the Octet allows earlier, faster and cheaper clone selection compared to the HPLC method. Test volumes are small, 100 microliter, which can also be reused. The assay time is shorter. You can measure as many as 96 samples in 30 minutes. And what I also believe is an advantage is that the extinction coefficients are not needed in this assay. Next slide. Next, I would like to show some examples of how the Octet can be used in the early research phase in which the objective is to discover broadly neutralizing antibodies that can be used in a cocktail for antiviral treatment. So how can the Octet be useful? The Octet can be useful by testing the binding of antibody leads to different viral antigens, to test the breadth of binding, to assess the binding kinetics, binding rates, or antibody maturation, for example, to test the competition between antibodies that need to be into a cocktail, quantitation of recombinant antigens, tagged or non-tagged. The Octet can also be helpful to learn more about the mode of action. Next slide. The first example I would like to present is from our influenza program. It is about the way binding kinetics supports the selection of broadly neutralizing flu antibodies. Next slide. Typical experimental approach is depicted in this slide. Due to the pH instability, hemagglutinin molecules are first biomolecular, followed by immobilization to stratification sensors. Next, association and dissociation of Fabs to the HA molecules is measured. The advantage of this approach is that the biomolecular HA molecules can be reused. Next slide. In this way, binding properties of antibody candidates to a large panel of HA molecules from different influenza strains has been assessed. In this slide, an example is shown of lead candidate antibody 01. It shows broad binding specificity to different flu clades. Next slide. An overview of binding specificities of 2 lead candidates has been published last year in Science. Based on the dissociation equilibrium data, it's clear that antibody CR6261 has a preference for group 1 influenza and CR8020 for group 2 influenza. Next slide. Apart from binding strength, also other information can be obtained using the Octet. For example, about the mode of action of our antibody candidates. In the current example, fusion mechanism of the flu variant to the target cell was mimicked on the sensor. This was done by immobilization of biomolecular H3, followed by trypsin cleavage of HA, subsequent binding of antibody 8020, lowering of pH to induce conformational change, followed by reduction with DTT, remove the disulfide bridge between the head and the stem region of HA, resulting in dissociation. In the last step, presence of HA head was tested using anti-head antibody. Next slide. The results of this conformational change assay is depicted in this slide. In the first step, HA was bound to stratification. In step 2, HA was cleaved by trypsin. Note that there is no effect on the binding response. Next, antibody was bound, followed by a pH shift and the DTT treatment. Already in step 3 was observed that CR8020 dissociated at the lowest pH. As a result of this, the head and stem parts of HA started dissociating. This was confirmed in the last step using an HA head binder. Whereas at a higher pH, much less dissociation was observed, indicating that 8020 actually prevented a conformational change of HA, thereby inhibiting fusion of the virus to the target cell. Next slide. Another important application of the Octet is to assess whether antibody candidates compete with each other for the same epitope, which is important to act in a cocktail. The standard approach we use to assess competition is the so-called 2-step blocking assay. In this approach, the antigen is first immobilized to the sensor, followed by binding of the blocking antibody, and next transfer of the sensor to a buffer containing the competing antibody in the presence of the blocking antibody. Test whether the antigen is blocked. Next slide. In this slide, some examples are shown of competition binding curves of antibody 01. In the left upper panel, binding responses are shown of antibody 01 to a non-blocked antigen. In the right upper panel, it is shown that the binding curve of antibody 01, the antigen blocked by antibody 2, comparable to the non-blocked antigen. Therefore, it was concluded that antibody 01 and antibody 02 do not compete for binding. This in contrast to the two lower panels, in which clear competition is shown for antibody 01 with itself and with antibody 03. For the analysis of competition, usually final binding responses are plotted. An example plot is shown in the next slide. From this competition plot, it is clear that antibody 01 competes with antibody 04, itself and antibody 07, not with the other tested antibodies. In as little as 20 minutes, competition between 6 antibodies can be measured using the Octet. Next slide. The next example is about competition experiments on HBs antigen, the envelope protein of hepatitis B, which is expressed by hepatitis B infected cells as a 20 nanometer sphere. The HBs antigen is a multivalent vesicle and is also used in our hepatitis B vaccine. Competition between HBs antigen-specific antibodies was first assessed using the two-step blocking approach using biotinylated HBs antigen. Next slide. However, the two-step approach did not result in conclusive data because for some reason the binding capacity of the antibodies differed largely amongst each other, resulting in very low and very high saturating binding responses. For this reason, it was decided to change the orientation by mobilizing the antibodies to streptavidin sensor, followed by binding to HBs antigen. This way, the binding response became equally high between the different antibodies, allowing one-step competition experiments. Next slide. An example of the one-step in solution competition is shown in this slide. From the dose-response blocking with antibody mAb01, it was clear that antibody 01 competed with itself and the other 3 tested antibodies. Next slide. The last example of this presentation involves the measurement of binding of an intact virus, influenza in this case, to the sensor. 2 capture strategies were followed to capture the flu variants. One was using a lectin, DNA, and the other was the use of an HA binding antibody. For both sensors, virus binding was observed, and remarkably, virus binding responses were negative, which is probably caused by the large size of a virion, which is about 100 nanometer. Such a large size apparently induces phase shifts that are larger than 1 wavelength. Next slide. In the analysis software of the Octet, negative binding curves can be flipped, converted from negative into positive binding, which makes the interpretation of such data easier. Also, we have some indications that binding of antibodies on top of the captured virions is possible, but this needs some more investigation. Next slide. I would like to end this webinar by summarizing the main applications and observations we did for the Octet. At Crucell, the Octet platform supports early antibody discovery by generating high-throughput information on binding kinetics, competition, and other functional binding properties. Several quantitative assays are currently used or in development. In our hands, particles with a diameter as large as 100 nanometer, such as viruses, can be analyzed on the Octet. Notably, if vesicles exceed a certain size, somewhere between 20 and 100 nanometer, binding responses become negative and need to be flipped. Next slide. Finally, I would like to thank my colleagues who contributed to the data of today's webinar, and of course, the people from ForteBio for their help. Back to you, Tamlin.
Thanks, Maren. Before we move on to our next speaker, I want to remind you that you can submit questions at any time by typing a question into the Ask a Question box on the lower left of your screen and hitting Submit. Our second speaker is Mark Fisher, professor in the Department of Biochemistry and Molecular Biology at the University of Kansas Medical Center. Mark will talk about the development of a high-throughput assay to detect protein folding and unfolding events using chaperone proteins and BLItz technology. Mark, you can begin now.
Thank you, Tamlin. I would like to thank ForteBio for sponsoring this webinar and inviting me to present some of our results using the Octet and BLItz systems. Today I'm going to present our development of a novel screening system that uses chaperone proteins, which detect protein folding and unfolding events. The goal of this screening system is to identify potential protein stabilizer lead compounds. Next. The problem that our lab wants to solve is to develop a screening system that will rapidly identify protein stabilizers or correctors which will bind to proteins that are inherently unstable. Some of the most relevant proteins that exhibit these properties are mutant proteins that result in protein folding diseases. As shown in this reaction diagram, current screening methodologies that identify potential correctors of the fold are often constrained by the inherent aggregation propensity that these systems exhibit. Thus, proteins that have a tendency to show aggregation or misfolding that arise from multiple states are kinetically unstable and are, in reality, moving targets. Next. In vivo, aggregation propensities are diminished by special proteins, commonly referred to as chaperone proteins, that are present in abundance and to prevent general misfolding events, primarily functioning by unfolding misfolded protein. These chaperone protein networks have been shown to be absolutely essential for maintaining protein homeostasis within the cells. Next. One of the most important chaperone protein classes in the cell belongs to the chaperone protein system. This chaperone is highly promiscuous because it interacts with a huge volume of partially folded proteins, perhaps explaining why it is an essential protein in vivo. This protein machine has a very complex cycle driven by ATP hydrolysis, illustrated on the right. If ATP is removed from the chaperone, one gets a platform that has a very high binding affinity for practically any protein that exists as a set of dynamic partially folded conformers. The binding strength of the high-affinity ATP-free chaperone often approaches affinities observed in antibody-antigen interactions. In vivo, this binding affinity is reversed by ATP binding and hydrolysis. In this talk, we will illustrate how we can use this high-affinity chaperone in our novel ligand screening platform while avoiding general aggregation. Next. Our use of this form of the protein comes from observations reported by George Larmer's group. They specifically noted that the high-affinity chaperone could potentially bind to partially folded proteins that were in dynamic equilibrium with their folded forms. As shown on the left, they observed that the activity and free concentration of the protein dihydrofolate reductase, or DHFR, decreases with time when a high-affinity form of the chaperone was also present in solution. This binding was reversed when ATP is added. The seminal observation that is pertinent to my presentation today is that the DHFR binding reaction onto the chaperone can be prevented when stabilizing ligands are present. This early experiment is the basis for our expanded use of the chaperone system, where we will use this system to identify protein ligands that bind to a target protein, thus shifting the dynamic equilibrium toward the native stable fold. Next. We have repeatedly found that folding disease proteins that naturally equilibrate with partially folded forms readily bind to the high-affinity chaperone. We use this chaperone sync technology to develop a simple readout that identifies potential protein stabilizers. In this new reaction diagram, the decision that a potential corrector or stabilizer of the native fold is present simply depends on the observable increases or decreases in chaperone binding. For example, if a lead compound candidate does not stabilize the target protein in question, the chaperone will bind to this protein in a time-dependent manner. If a stabilizing ligand is present that binds to the native fold, the chaperone binding reaction will be diminished. It is a simple yes-no decision point. Next. We have incorporated this chaperone screening platform onto the ForteBio Octet label-free biosensor system. We developed two complementary setups with the Octet system to detect protein-stabilizing ligand. In the first setup, we can attach a target protein of interest onto a Layer Interferometry tip or biosensor in a manner that extends the target protein away from the surface. We then dip these labeled tips into solutions containing first the lead compounds alone, and then move these tips to wells containing the chaperone and the same lead compound candidates. In panel A, if a ligand fails to stabilize the attached target protein, the chaperone binds and produces a large signal due to its large size, in this case 802 kilodaltons. In panel B, if a stabilizing ligand is present, the chaperone binding reaction is prevented or diminished, resulting in a significant decrease in signal. We can rule out false positives due to the chaperone inhibition by using internal controls using proteins that exist as partially folded conformers. Next. In the second setup, we can attach the purified chaperone onto the tips and observe if the target protein binds to the chaperone or is stabilized by potential lead compounds. In both instances, we can generate an easily observable yes-no output signal that allows us to identify if a lead compound is a potential ligand or stabilizer. Next. In the next series of data slides, we demonstrate the utility of this novel chaperone platform, where we use this system to identify potential protein binding compounds that may be stabilization candidates of the nucleotide binding domain of the cystic fibrosis transmembrane regulator, or CFTR-NBD1. This protein was generously supplied by the Cystic Fibrosis Foundation. In the second instance, we show that we can detect when a natively folded protein can be stabilized by known ligands. In the third instance, we demonstrate that we can detect when a protein is properly folded by examining the chaperone binding differences between folded or partially folded molten globule forms of that same protein. In the last instance, we demonstrate that we can observe the large-scale unfolding and refolding transitions of the anthrax toxin pre-pore to pore transition when this bacterial toxin component is attached to the Layer Interferometry tip. We should note that all the examples listed in this data summary slide were initially tested and confirmed using surface plasmon resonance. In all instances, we can truly state that high throughput capabilities, particularly the ease of use and direct hands-on time of the ForteBio Octet label-free detection system, is less cumbersome and more rapid than more popular SPR technologies. Next. An example of the kind of targeted research that is currently being pursued with diseases that results from protein misfolding depends on the observation that one can achieve a stable fold of a mutant protein such as the CFTR-NBD1 domain, both in vitro and in vivo, when conditions are optimized to allow for the slow folding of this protein. The goal, then, is to identify appropriate pharmacological chaperones, small molecules that can also stabilize the native fold. The structure of the CFTR-NBD1 protein domain in question is illustrated in the lower left-hand corner. The most common disease mutation in cystic fibrosis comes from an amino acid deletion at position 508 that normally encodes for a phenylalanine residue. This deletion results in an abnormal folding and trafficking, resulting in a wide variety of phenotypic disease states that primarily affect lung and pancreatic function. This realization that the folding can be potentially corrected in certain protein folding disease states has led to the relatively new and recent therapeutic efforts to actively develop targeted pharmacological small molecule chaperones to specifically reverse these misfolding events. Next. As our benchmark experiment with CFTR-NBD1, we demonstrated that the signal from chaperone binding to the destabilized and tip-immobilized CFTR-NBD1 protein was significantly diminished when a known stabilizing ligand such as GTP was present in solution. Importantly, the chaperone exclusively binds ATP, whereas GTP fails to bind to the chaperonin. Thus, this diminished chaperonin binding results from the fact that GTP binding to the CFTR-NBD1 domain thermally stabilizes this protein, resulting in an increase in the correctly folded form of this particular domain. We next tested a small series of potential CFTR stabilizers, commonly called correctors, that were generously supplied by the Cystic Fibrosis Foundation, and we used these compounds to screen for potential direct stabilizers. Next. In a typical screening assay, we found that 3 of the 4 candidates tested in this initial panel, indicated by the stars, showed signals that tracked between the completely destabilized form of CFTR and the known stabilizer GTP. In general, compounds that simply decreased chaperonin binding are initially classified as potential hits. Next. From this initial set of 10 correctors, we found a total of 4 candidate compounds that showed potential for stabilizing CFTR. We subsequently found that 2 of these compounds had previously and independently been demonstrated by Phil Thomas's group at UT Southwestern to inhibit large-scale aggregation of the CFTR-NBD1 domain. Next. Most importantly, this slide shows that these potential stabilizing compounds that appear to stabilize the normal domain also appear to stabilize a mouse mutant containing the common phenylalanine-deleted CFTR-NBD1 domain. Although this single domain has previously been used as model systems for this protein folding disease, the current consensus in the field is shifting toward approaches to use the complete transmembrane regulator protein as a model to search for protein pharmacological chaperones. Nonetheless, our experiments still validate our approach for rapidly identifying potential stabilizers of misfolded proteins. Next. Versatility of this chaperone and capture system to identify potential ligands for non-disease proteins or just to verify folded states are exemplified in the next 2 slides. In both instances, chaperonin was immobilized on the tip, and we simply determined if the chaperonin could bind the protein of interest in a time-dependent manner. In the first case, the older experiments of Lormer's groups were simply recapitulated, and these experiments clearly demonstrate that we can rapidly identify naturally occurring protein-binding ligands that result in the stabilization of this protein. As seen previously, the presence of stabilizing ligands results in a marked decline in signal due to the decrease in DHFR's binding to the immobilized chaperonin. Next. Furthermore, the next slide demonstrates that we are able to detect if a proper fold has been obtained by comparing folded with partially folded forms of the protein alpha-lactalbumin. We can detect if a significant population of the protein has acquired a proper fold. As one can easily observe, there is a dramatic signal decline in protein binding to the chaperonin when the protein has adopted its proper fold. Next. The examples presented are just a small sampling of the wide variety of protein systems that we now can use to identify protein stabilizers using our highly pure chaperonin system under near physiological conditions. In our laboratory alone, we have documented over 50 proteins that interact with the chaperonin as partially folded proteins. 11 of these proteins are classified as authentic folding disease proteins. In all cases, we can readily observe chaperonin binding to these moving targets, and now with the Octet system or BLItz system, we can rapidly interrogate folding integrity of any protein that exposes a hydrophobic surface to solvent, even if this occurs transiently. Next. Comparison of the ligand screening protein stabilizer systems indicates that our particular chaperone platforms allows us to identify stabilizers for transiently stable proteins, something that other current biophysical systems will have difficulties with because of the inherent problems that accompany protein misfolding and aggregation. Using the chaperonin as our protein folding detection platform is a highly advantageous methodology, and a growing list of advantages are listed on the left-hand side of this slide. Next. In the last series of slides of this presentation, we also found that we could visualize a large protein folding conformational changes that accompany the unfolding and refolding transitions of the anthrax toxin pore translocon using the ForteBio Layer Interferometry system. These latter examples have also been confirmed and extended using the newest single-channel ForteBio system called the BLItz. This slide simply illustrates the transition that we are examining. In the endosome, under acidic conditions, the soluble form of the anthrax pre-pore protective antigen, shown on the left, transitions to a membrane-insertable form, shown on the right, to aid in the transport of lethal elements of the other two anthrax toxins into the cell. This transition of the pore structure results in an extremely large conformational change, from 85 angstroms to 180 angstroms. And this conformational change and this resulting structure has easily been documented in our lab using electron microscopy structural analysis. Kinetics of this transition, however, are extremely important to document, but real-time direct measurements of this transition have been difficult due to extensive protein aggregation. Next. We initially employed surface plasmon resonance to measure this transition, and indeed documented that we could view this transition when we specifically orientated the pre-pore conformer onto the SPR surface and examined the transition as neutral buffer was replaced with buffers that approached endosomal acidification conditions. This transition occurs rapidly, and once transitioned, the conformation remains locked in its pore form once flow is returned to the neutral conditions. Although successful, these series of SPR experiments were extremely tedious and time-consuming. Next. Both to our surprise and delight, we demonstrated that we could also view this very same type of conformational change using the ForteBio Octet or BLItz biosensor systems. In this instance, not only were we able to observe this conformational transition, we were also able to simultaneously view this transition at a series of pH conditions in duplicate. What would take us 3 to 4 days to complete with SPR instrumentation we use currently at KU Lawrence, we were able to accomplish within 40-minute time window, that is from immobilization to pre-pore to the transition pore form using the ForteBio Octet systems. We validated this pore formation by knocking the translocon off the biosensor tip and viewing the pore conformation directly on electron microscopy grids. Next. In conclusion, we can readily detect the appearance of kinetically transient yet partially folded states in proteins. The chaperone-independent biosensor signals can be generated using the target protein immobilized on tips or with a chaperone attached onto the biosensor chips. As with all transitions, we can kinetically control these systems by varying solution conditions. In all cases tested thus far, we have found that the ForteBio label-free systems are superior in high-throughput screening capabilities compared with the surface plasmon resonance label-free systems that we have used. Finally, much to our delight, we can even determine large-scale conformational refolding transitions, in this case using the anthrax toxin pore translocon, using the simple label-free systems developed by ForteBio. Next. I'd like to acknowledge my graduate student, Subhaschandran Naik, rotation students who have used the BLItz system and the KU Protein Production Center for allowing us to use their SPR system. Also from ForteBio, I'd particularly like to thank Wesley McGinn-Straub and application scientist David Appio for their help directly with this system, as well as the interaction that I've had with the sales and marketing team, headed by Chris Silva. Thank you.
Mark will be available at the end of the presentations to answer any questions you might have. So if you do have a question, please submit it now. Our third and final speaker is Jimin Yu, Senior Scientist at Didexis. She'll discuss a binding rate screen she developed to facilitate the identification of constructs suitable for recombinant protein production. Jimin, we're ready when you are.
Thank you, Tamlin, for that kind introduction. I would like to thank ForteBio and Jing News for inviting me to be a part of this seminar. It's an honor, and I'm very excited to share the binding rate screen with this audience. The binding rate screen is a high-throughput assay that allows one to quickly assess the integrity of the target protein while still insoluble cell lysate. Hopefully, by the end of this presentation, you will find this method to be a very practical tool that you could utilize to speed up the gene-to-structure process in your organization. Next slide. Here's the outline of my talk. I will first share with you some pre-binding rate screen experience, which really led me to have a strong desire to develop a novel method. I will then describe the principle behind the method, how it's done, and how we have demonstrated the method works. Due to time limitation, I will only go through one additional example with concluding remarks. And by the way, all of this work was done while I was still at Novartis, and the work has been published in Analytical Biochemistry in 2010. Next slide. My first project in the protein science group at Novartis was to screen through hundreds of constructs designed for a well-known, difficult-to-crystallize kinase and to identify candidates that would lead to crystal structure. Let's refer to this protein as protein 1. Implementation of mutations and truncations throughout the protein sequence is a pretty common gene-to-structure strategy, and that was the approach we used. Over 200 constructs, all his-tagged, were designed with variations in the KID and C-terminus domain, and soluble expression was the criteria used to rank order these constructs. The screening approach we used, again, was pretty standard, as shown in next slide. The his tag on all constructs allowed one to quickly purify the target protein on small scale using the QIAGEN system. The soluble lysate, IMAC flow-through, and the AUIT were then analyzed by SES page, followed by Western or dot blot. Typically, this process took about 3 to 5 days, depending on the number of constructs and FTE. In literature, there have been papers describing the use of biophysical methods such as DSC, NMR, etc., to screen the IMAC product. However, such methods usually require proteins to be relatively pure and that there is a large amount of it. These methods are also quite time-consuming. Next slide. Shows our first example. In our protein 1 example, the SDS page gel analysis of the crude soluble lysate and IMAC AUIT showed that as the C-terminus length increases, the soluble expression also improved significantly. Well, that was fantastic. Yes, we did notice that there was protein in the IMAC flow-through, but that could be just because the nickel column was overloaded. So we ended up picking the winning constructs for scale-up purification based on soluble expression. However, those purifications were always troublesome because the proteins were high maintenance. Even when we successfully made nice monomeric proteins, none ever produced crystals despite numerous crystallization conditions attempted. Next slide. It wasn't until we examined the IMAC flow-through one day to realize that the target protein in the flow-through was aggregated, suggesting that perhaps even if you could isolate the monomer with brute force, it's not of high quality. So we then asked the question, what is the quality of the target protein expressed? It became very clear that evaluating the oligomeric state of the target protein in soluble lysate was essential. We then implemented methods to look at the ratio of the aggregated to monomeric protein in soluble lysate using analytical sizing, chromatography, and electrochemiluminescence-based immunoassay from mesoscale discovery. This was a very labor-intensive process, but it got the job done. When we finally started to focus on constructs overexpressing high percentage of monomeric protein, not necessarily high soluble expression, we immediately had a success. Those proteins were easy to work with and behaved very nicely during purification. Next slide. Summarizes our in-house experience with this kinase campaign, which actually was done in 2006. The process involved screening through the constructs, identify trans, and select constructs for scale-up purification and crystallization. For this kinase, the process was repeated several times. In the end, it took 10 months and many FTEs to get the crystal structure, and that actually was considered a huge success because many labs, both industrial and academic, had tried and failed getting the structure over several decades. And the valuable lesson we learned was that improving soluble expression was not equivalent to having access to quality protein, since you can have soluble aggregate. At that time, in 2006, most published literature were describing high-throughput methods designed to screen for improved soluble expression only. There were also papers describing biophysical methods to look for quality protein. Again, these methods typically require a large amount of clean protein, which means it's not truly high throughput. To me, this was obviously not the way to go, and there was a need for a new assay to be established which can screen for soluble aggregates, ideally in crude lysate, to save time. And to be able to do this, you need a system that will meet 3 criteria: that it needs to be able to tolerate crude lysate and needs to be able to specifically identify your target protein from many other proteins present in the crude lysate, which means the method needs to be antibody-based. And lastly, the measurements need to be done in a high-throughput fashion. To my knowledge, no method was able to do that at the time. Next slide. One day I was sitting in a ForteBio seminar at Novartis, and I was blown away by the technology. I assume most of you are familiar with this technology, so I'm not going into details. It is sample matrix friendly, meaning it could do measurements in crude soluble lysate, media, serum, and it's high throughput, capable of doing A measurements in parallel. Now, of course, you have the 16-channel option, and you have easy access to fresh sensor service at a reasonable price. And lastly, the instrument is simple and robust. All you need are sample, sensors, and plates, and you're pretty much ready to go. I thought this was one of the coolest technology, and it certainly met the 3 criteria that I mentioned previously. Next slide. Slide 9 describes the idea that I had in mind for the novel assay. In almost all high-throughput screening projects, an affinity tag is always engineered into the protein construct for purification purpose, and the his tag being most common because of its small size, and it's been well documented, studied, and widely used. Here are 5 constructs. All are variants of the same parent protein. The red block represents the his tag at the end terminus. Assuming these are the conformations of the soluble expression upon expression, then you would want to avoid construct number 4 because of aggregation, and construct number 5 because the tag is hidden and would not be available for purification. Construct 3 and 2 are favorable, and not sure about construct 1, probably not ideal. And the beauty of the his tag is that it not only serves as the purification handle, but now every construct has this identical stretch of a sequence, which can then serve as a probe to sense the microenvironment of the target protein. And there is good antibody to this tag that's available commercially, and if the tag is free and available, then it will bind to the his antibody on the biosensor. If not, then it won't. Next slide. So the principle of the binding rate screen is very simple. What we're interested in measuring is the initial binding rate, which is directly proportional to the binding rate constant and the concentration of the his tag target protein. The binding rate constant describes the inherent ability of the his tag on each construct to bind to the anti-his antibody. In the hypothetical example provided here in this figure, the binding rate constant of construct 3 and 2 is expected to be greater than that of 1, followed by construct 4 or 5. The goals of most gene-to-structure screening projects are to identify constructs capable of producing sufficient monodispersed proteins that are biologically relevant. While the binding rate screen cannot screen for the latter, cannot screen for the activity, it certainly can for the first 2 agenda. The faster the binding rate, the more desirable the construct. Constructs with high binding rate constant and concentration in crude soluble lysate would have a high value binding rate, and they would rank order high, followed by high binding rate constant with okay concentration. because you're still getting protein with high quality. Low binding rate constant, however, would give the construct a low ranking order regardless of the concentration of the soluble protein. And lastly, extremely low concentration of soluble expression would also give the construct a low ranking as well, regardless of its binding rate constant. When in doubt, you can always run an SDS gel to verify soluble expression level at the protein size. It is very important to mention that we are always comparing variants of the same protein in each project. Only then the change in the binding rate constant reflects the impact of modifications made in the protein sequence. And different parent proteins have different binding rate constants, so you can't compare them directly. Next slide. The protocol to run the binding rate constant binding rate screen is very simple. Preparation of the soluble lysate is pretty standard. You can lyse the cells with preferred lysis buffer, and the cells can be E. coli, baculovirus, or mammalian. After centrifuging the lysate, the soluble lysate can be filtered if necessary. The crude soluble lysate can then just be diluted with sample diluent from ForteBio. For the biosensors, you can either purchase the pre-made anti-His antibody-coated biosensors or make your own to save the cost. The penta-His antibody from Kaigen works the best in our hands, and you can coat the sensors following the ForteBio protocol, and they can be stored dry for at least 6 months. Once you have the protein samples and biosensors, you can run the assay in quantitation mode. Because we're only interested in initial binding rate, less than 5-minute measurement time is more than enough for a column of 8 samples. And you can find all the details in our publication. Next slide. With the huge amount of historic data on protein 1, we had a perfect opportunity to test the binding rate concept. The image on the left is the SDS page gel of crude soluble lysate from 8 constructs that are representative of the constructs that were chosen for scale-up purification. The last column is the soluble lysate from a negative control construct that does not express His tag protein. The image on the right shows the IMAC elution profile, and I can tell you now that it was a construct E and F that gave crystals immediately, and the structure of F was solved. Next slide. We then used a sizing chromatography and immunoassay to estimate how much monomer protein was present in crude soluble lysate for each construct. Basically, the soluble lysates were fractionated on sizing column, and A280 profile is shown in black. Fractions were then collected, and each were examined by ECL-based mesoscale diagnostic technology with anti-His antibodies to look for where the target protein was eluding and how much there was. The ECL signals for each fractions are graphed in red in the top example figure and black in the bottom figure. As you can see, construct A, B, G, and H all contain mostly aggregate soluble target protein. The ratio of monomeric to aggregate protein improved significantly in construct D, E, and F. But there really wasn't that much protein in D. As I mentioned earlier, construct E and F were the winning constructs at the end of our 10-month-long project. Next slide. Next, all crude soluble lysates were screened by the binding rate screen, and as you can see in the middle panel, within 1 minute you can identify the most promising constructs to pursue. It was extremely exciting to see that construct E and F had the highest binding rates, which were calculated using the linear data from the first 10 seconds. The binding rate value certainly makes prioritizing very easy. If you want to figure out why the other constructs weren't ranked high, you can always run a gel to do additional analysis, such as analytical sizing. However, if you don't have the time, at least you know what would be a good choice to scale up first. Next slide. We further went on to demonstrate that each construct variant of the parent target protein indeed has its own binding rate constant. Monomeric protein of selected constructs were screened at the same final concentration, which is 0.3 micromolar. If the His tag on all these variant proteins were equally accessible, then you would expect to observe the same binding rate for all constructs. However, this was not the case clearly. Again, construct F, whose structure was solved eventually, excelled. Interestingly, construct D excelled as well. In fact, construct D was put into trees previously as well and had given crystals, but the overexpression of this construct was extremely low, and a huge culture had to be grown in order to isolate enough material for crystallization. This was why construct D ranked low in the binding rate screen. Had we known that during the actual study, such construct would have been deprioritized, since there were other constructs such as E and F that gave similar quality protein but overexpressed at much higher level. Next slide. In the second example, 30 constructs of a DNA-binding protein, referred to as protein 3 in the paper, were engineered and screened. Again, all constructs contained his tag, and truncations were made at both the N and C terminus. Gels of soluble expression and IMAC elution revealed that construct B4 through 5, C4 through C5, E3 through E5 were good candidates, but it was rather difficult to choose the best one based on these gel images. When the crude soluble lysate of all 30 constructs were screened by the binding rate screen, which took only 20 minutes, that is 5 minutes per column, which holds 8 samples, the ranking of the constructs became very clear. B4 was the best by having the fastest binding rate. In fact, it was B4 that gave crystals, and its structure was solved. Next slide. Analysis of crude soluble lysate from the B series constructs by sizing and ECL immunoassay revealed that with protein 3, soluble aggregate was not an issue with any of the constructs. B4 simply contained the most amount of monomer protein. So again, had we had the binding rate screen during the study of protein 3, we could have reached that decision to prioritize construct B4 within 20 minutes instead of trying to follow up with all the other constructs that look good by gel. Again, this method could have saved us a lot of research time and resource. Next slide. So in conclusion, I hope I've convinced you that the binding rate screen is a novel functional screen that's very useful and uses the his tag as a reporter and measure its binding rate to an affinity matrix as a metric to reflect aggregation, concentration, and purifiability of the target protein. The method is built on the ForteBio platform, which is very robust. It is the instrument is sample matrix friendly, it's high throughput, and very easy to use. And the cost of each biosensor is expensive, but I do believe you save much more on FTE and resource. And since 96 constructs can be screened in 1 hour or less, so the method is very fast and easy to use. I would like to say that the value of the binding rate screen to structural biology efforts of rapidly generating and characterizing truncations is to prioritize protein variants so that the constructs best suited for crystallography can be put into trials, first to improve the chance of individual target success, and it does not guarantee that you will get crystals and structure. Next slide. Finally, I would like to thank my Novartis colleagues who worked with me on this project. Dr. Storlick gave me tremendous help in fleshing out the idea into a manuscript. Gwen and Arun provided cultural materials, insights, and enthusiasm. Ying, Wei, and Emma were very helpful in carrying out the traditional analysis. I would also like to thank my former boss, Isabel Zurl, who gave me the opportunity to follow my ideas and gave me tremendous support. I'd like to thank ForteBio for their tech support. I'm currently at Didexis. If you have any questions, please feel free to reach me at this email address, and I thank you for your attention. Back to you, Tamerlan.
Thanks, Jimin. Before we begin the Q&A session, I want to encourage you one final time to submit questions for our panelists. Simply type the question into the Ask a Question box and hit Submit. All right, so we're ready to start our Q&A discussion. We have a lot of really good questions, and we'll answer as many as we can. Our first question is for Maren, and the question is, in the kinetic fits, which model works the best or is recommended?
Yeah, thank you, Tamerlan. Well, I think that is a kind of general question, and does not only apply to the BLI technology, But what we, in our opinion, we always try to use one-to-one fits. So that means that we need monovalent antigens or binding molecules used as analytes, such as Fab. So if we have an antibody, we always try to get the Fabs to measure in the end the one-to-one binding to the antigen. And for the fitting, we use the one-to-one model, and that can be either the partial fitting or the full fit model that is supplied by the ForteBio software. And in general, that's the best way to get the individual binding rates, from which in the end you can also calculate the equilibrium dissociation constants.
All right, Marin, now I have some slide-specific questions. On slide 24, the person says it was mentioned that no change in response was on trypsin treatment. Shouldn't there be a change in response since there is a change in mass due to cleavage?
Yeah, well, to this question I can be short. There is no mass difference because it's only a cleavage. So there's only one peptide bond is cleaved by the trypsin, and there is no net mass change. Yeah, well, maybe if you measure it on a mass spec, but there's essentially no mass differences, and that's also what we observe. There's no differences.
Okay, another one. This one is on slide 20. How many times can immobilized tips be reused, and may tips be preserved in their running buffer, and for how long?
So, I think this question relates to our hemagglutinin immobilization. Unfortunately, this is an example of quite an unstable antigen. And that's the reason that we, in the end, did not use or were able to reuse the tips. And therefore, we were happy to work on the Octet because it's easy to use or to dispose the sensors and to reload the antigen for a new run. And actually, we started with another SPR system, and we actually did not manage to get the regeneration working. So we were happy to perform these experiments on the Octet, where you can measure as many as 8 or sometimes 16 tips at the same time. But in general, you can say that you can preserve the tips in a running buffer for as long as your antigen is stable.
Okay, Mark, I have some questions for you now.
Yes.
The first one is, wouldn't the immobilization of protein of interest onto the optics sensor probe change the native conformation of the protein, i.e. in solution versus immobilization?
So if you looked at the particular slide presentation here, one of the things that we had done to avoid that particular problem, we don't attach the proteins directly onto the tip surface. In most cases, we have the protein attached through a chimeric construct. So if you have a GST construct, for instance, one can position the protein that's away from the immobilized tip. That's one of the problems we had run into early on. If we just immobilized onto an NHS type tip surface, mean reactive surface, we would run into that problem. So we have to actually extend the protein from the tip surface. We can also take advantage of situations where we can raise antibodies to the particular protein of interest and use an antibody sandwich with a anti-FC antibody, a tip from the Octet system, and then subsequently bind the antibody with the protein of interest. We did that with the CFTR protein. Also, we can it turns out we can also immobilize the chaperone on the tip and interrogate the protein directly in solution. So that problem is avoided in that particular case where you would have the changing in the native conformation of the protein.
So that's always a concern, and that's a very good question, and that's one of the reasons why we have two different configurations that we use.
Hey, Mark, can this Chaperone be used for any protein type?
It turns out that the Chaperone is a very promiscuous protein. If you have any protein that has a hydrophobic surface that's exposed, transiently exposed, and so forth, it will bind almost any particular protein. The proteins that we are refractory to the Chaperone binding are those that are actually natively folded, or in some cases are very hydrophilic. For instance, you have multiple phosphorylated proteins, and then, for instance, tau can't bind to the Chaperone. If it's heavily phosphorylated. But this is one of the both the boon and the bane of the Chaperone system, is that one can actually bind proteins very easily. And one of the early purification schemes that were generated unfortunately pulled out a lot of unfolded proteins along with the Chaperone protein. So we have developed a particular rapid protein purification scheme that's actually better than what is commercially available. And we avoid, so we actually get a clean Chaperone, and this can be used for any protein that undergoes a partially folded state and exposure of hydrophobic patches.
Okay. Mark, for the Anthrax experiment, did you immobilize membrane containing the translocon onto the tip?
For the Anthrax experiment, we actually immobilized the pre-pore onto the tip via an affinity attachment of the lethal factor onto the tip itself. So the pre-pore was binding in a directional manner, and the transitioning of this pre-pore to pore it was early. We initially observed this with some early experiments that we published in Nature Structure Biology in 2008, where we could actually observe the EM structure of the anthrax toxin pore. In this particular case, we did not have to have any membrane present to observe that. We've recently, so we didn't actually attach the membrane per se. We were just actually transitioning this pre-pore to pore directly on the tip.
Okay, Mark, the final question for you is a listener was wondering if he understood correctly that you were able to remove protein from the sensor surface and use that for EM.
Yes, that was both a surprise and because the reason why we can do that is we can actually remove the protein from the tip, because we've attached the lethal factor via a disulfide linkage. We simply treat with DTT and remove the protein from the tip directly onto the EM grid, and we were able to observe the conformation that we routinely observe when we're doing reconstruction of the anthrax toxin pore translocon using electron microscopy. So we had the advantage of actually knowing what we were looking for, and indeed, we know what an aggregate looks like, and we know what a pre-pore looks like, and we know what a pore looks like. So we could definitely pick out the pore conformation.
Thank you very much, Mark. G-Man, I have some questions for you. The first one is, other than the examples provided in your presentation, have you had good correlation between the ForteBio results and the crystallization success in general?
I would say the answer is yes. I think subsequent to the publication, we've tried this method in probably like 10 or so projects. Of course, a lot of this is also always the screen was done for projects that already you know what the answer from the biochemical screen is. In most cases, the results correlate very well with the constructs that are picked out by the traditional biochemical analysis, and these are the constructs that will give you either crystals or they match the results from biochemical studies. And there are a couple instances where it proposes a different construct candidate. However, in those cases, because you don't know what the result were, because those weren't selected in a general in the traditional screen method, so they weren't characterized. So the binding rate will suggest that something else might work better. But overall, we say yes, the method works quite well. And it's very fast.
Does your method work with NTA sensors?
I think very early on when I was trying to develop this method, I did try I looked and thought about the NTA because you can strip it and then recoat the sensor. I did not try this, but another colleague at Novartis did this, and then I think his comment was that it wasn't working quite as well as the anti-His antibody because the antibody was giving better sensitivity. So I don't know if I don't know how many different NTA material that he has tried, but maybe there are material out there that you could use to try to see if it works. Yeah, but I think it should work if you can find the NTA that will give you good binding characteristics.
All right, we have a listener wondering if this system function is just for aggregate proteins.
I think the system, the goal was to actually screen out the protein constructs that will generate aggregate protein so that you can continue to work with constructs that are expressing good quality protein. So it's not just, well, I guess it is to screen out the aggregate, but that's not the only function of the assay. It also tells you which construct that you want to move forward with, which one has the best soluble expression, not just the soluble expression, but also good quality protein that you can isolate right away. You can get that. So in other words, even if the proteins are not aggregated, like the example 2 that was provided, that you can pick out the one that was expressing the most amount of the soluble protein and move forward with.
Great. I have one final question for you. What do you expect is the net savings using the ForteBio method versus traditional screening, given that the sensors are expensive, and can the sensors be reused?
That's a great question. So the way I looked at it is that, say, if you have 100 samples and each of the sensors, if you coat your sensor yourself, which is the sensor costs about $4, and then you spend a couple $100 buying antibody, the anti-His antibody from Kaigen, and which for 100 for I can't recall the amount, 100 micrograms or so, that you can actually use that for quite a while. So each sensor is $4 for 100 constructs. That would cost about $400. Let's say if you want to repeat the assay, that's $1,000 versus. So you know the answer in a couple hours versus you have 3 or 4 people running expression gels or running analytical studies, go through high throughput purification and running everything on gels and sizing column. So calculate the cost for that, and I think the binding rate screen is actually, to me, it's a lot more economical. And the sensors, I have not tried to reuse the sensor because actually the cost wasn't that bad. To us, it was already saving a lot of money. Yeah.
Wonderful. Thanks, Jim.
Can I actually also address that regeneration aspect? So this is Mark Fisher. We've also found that we could reuse Chaperone and tips very easily because of the fact that you can actually reverse the binding affinity by adding osmolite ATP mixes, and then you can regenerate the tip with the chaperone on there. And also with an attached protein, if you have the chaperone bound, you can simply dissociate that chaperone from the bound state with the ATP osmolite regeneration system. So it's a very physiologically safe and useful method for reversing the binding.
Thanks, Mark, for weighing in on that. Our time is up. Thanks for listening, and thanks for the great questions. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank our panelists, Maren, Mark, and Jimin, as well as our sponsor, ForteBio, a division of Paul Life Sciences. We'll be sending out a survey shortly and hope you'll take the time to respond. Your comments help us continue to provide topical and timely webinars. Thank you.